So hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're so glad you're here today. And we will be talking about academic drug discovery on two specific projects, kidney diseases and Duchenne muscular diseases with Donna Huron today. Welcome to the show, Donna. We're glad to have
Thank
you.
you. Thank you. It's great to be here.
Donna Huron began her career as a medicinal chemist in the pharma industry, working at Hoffmann La Roche and at Wyeth Research. We just briefly talked about this. In that capacity, she actually contributed to drug discovery efforts for HIV, cancer, asthma, and CNS disorders with an increasing amount of responsibility there. In 2004, she joined academia, where she helped establish drug discovery and medicinal chemistry centers both at the University of Pennsylvania and at the University of Pittsburgh. She was a professor at the University of Pittsburgh School of Pharmacy until 2023, when she joined the chemistry department at the University of Pennsylvania. As we will learn today, her laboratory focused on the design and synthesis of small molecule probes and drug candidates as well to treat cancer, kidney disease, and genetic diseases as well. Donna is a fellow of the American Association for the Advancement of Science and the ACS. And Donna was also inducted into the ACS Medi Hall of Fame in 2023. In addition to that, she received numerous awards, among others, the ACS Philadelphia Local Section Award, the Philip S. Portuguese Lectureship, and the Garvin Olin Medal. Donna held a number of roles and elected positions at the national level in the ACS and the AAAS, and is the co-author of the textbook Medicinal Chemistry in CRC Print. From 2014 to 2023, she was also an associate editor of the ACS Medicinal Chemistry Letters. And in 2019, she actually co-founded the Empowering Women in Organic Chemistry. And with that, in that, she still serves today on the organizing committee. So Donna, what an impressive CV. We're so glad to have you. And yeah, really an honor to have you here today with us.
Oh, thank you. It's really exciting to be here for me. So I appreciate the invitation.
Awesome. Yeah, and in our good old tradition, we typically start with some warm-up questions. So Donna, can you maybe tell us a little bit more about what inspired you to start a career in drug discovery?
I remember this day distinctly. I was in graduate school. At the time I was in graduate school, the sort of pinnacle of things you did was total synthesis of complex natural products. And so that was the what everybody wanted to do, what we were doing. I worked with the late Professor Amos Smith, and I was a little, we had made these compounds, you get an NMR and you write a paper. And I got my final product, and I was like disappointed that was that was it. And I remember going to a seminar that Noel Cohen gave. He was at Hoffmann La Roche, and he talked about arachidonic acids, these incredibly complex, really super sensitive natural products, and finding antagonists of the enzymes in the arachidonic pathway. And I remember going to that seminar. I remember what he talked about. And I remember the feeling leaving it saying, that's what I want to do. I really want to make molecules that really have an impact and do something, not just put it in a vial, take an NMR and publish it. And so he was my inspiration.
I love that. Yeah, I think many of us have like kind of inspiring mentors or inspiring talks, and like you as well, I had that same feeling too. Like during grad school, we often made compounds that we just washed the vial with acetone and poured it away, right?
Yeah. Yeah.
Uh, all right. Donna, can you talk a little bit about your favorite molecule and why it is your favorite molecule? And I think you have a slide on this,
I
right?
can, and my favorite molecule is calicheamycin. And I put a slide up because I don't know how familiar people are with it. And so let me share it. Can you see calicheamycin?
Yes. Incredible
Right.
molecule.
It's an incredible molecule, right? And so I remember also going to a Gordon conference where they talked about it's Natural Products Gordon conference, and the people who were then at Letterly described their isolation and characterization of this molecule, right? And look at this molecule. It has right? This basically a hexasubstituted aromatic rings. It has these really unusual deoxy sugars. It has a trisulfide. It has this NO connection, and that's not even the most interesting part, right? It's got a bridgehead olefin, but it has this enediene, right? And it basically gets reduced. You have a Michael addition here, right? That brings that enediene close enough together to undergo a Bergman cyclization, and it basically intercalates and alkylates DNA, right? And so, and importantly, calicheamycin was incredibly toxic, but it was the first payload for an antibody drug conjugate. And this was probably 20 years ago, and that sort of area has resurged recently, right? Now everybody's doing ADCs, but this was the first one. And so it's just such a cool molecule. I always tell my students, when you see a drug molecule, you should look and say, why is this fluorine group there? What's it doing, right? Why is this molecule here? So I look at this molecule and think, I wonder why nature put, a hexa-substituted phenyl group, on this. Why was that important? So it's kind of inspiring.
Oh yeah, I mean this, Donna. I think this is easily the most complex favorite molecule that that we've ever seen here on Drug Hunter. I mean this thioester there too. Yeah,
Yep.
we could go on and on about the unusual functional groups in this thing. Yeah, really very very impressive. So thanks for sharing that. I didn't know the structure of that, and the history is cool too with the ADCs that you
Yeah, yeah, yeah.
uh, cool. So, can you talk a little bit about what you're most excited about in drug discovery today?
Absolutely. And so it's kind of related to calicheamycin. The structures of the drugs that, that are coming out now are really exciting, right? I think medicinal chemists we can really be more creative, because now we have confidence that if we make a much larger molecule with unusual functional groups, they could still be active. They might become drugs. And so I think that's what I'm excited about that. And chemists usually had a very small playground, right? Small molecules, but now, we work on these much bigger molecules, PROTACs, right? Or ADCs even. So I think we have medicinal chemists have impact much wider than we used to have.
Oh yeah, I mean I couldn't agree more with that. I mean you have all these macrocyclic peptides these days,
Yeah.
and the protacs that you mentioned that are orally bioavailable. Some of them even CNS penetrant.
Yep.
So
I
yeah,
mean, and just
really.
think about these oral GLP compounds. Like, holy
Oh
mackerel.
yes.
Like, who would have thought? Like,
Absolutely. Yeah,
so,
yeah, yeah. Definitely like one of the, yeah, a really, really cool field. That's seems to give Lily an edge right now. So it's looking really, really cool.
yeah.
Stuff that we probably wouldn't have thought about a few years ago even.
Yeah.
Um, if you could give yourself maybe or the younger folks in the audience some career advice when you started your career, what would you say to your younger self or to kind of the folks that are just starting their career in medicinal chemistry?
So I think one of the things I should have done is taken more risks, right? Tried things that might, didn't have a perfect chance of working, just because, what this showed us is that a lot of the lore that we know as medicinal chemists may not be true, right? And so you could kind of try to push the boundaries a little bit. So there's a lot of things where I think I was more conservative in the things I did, the compounds I made. And I think, if I did it again, I would try to take a little more risks.
Yeah, I think that's good advice. You got to balance it though. You can't put all your eggs in the high
Yes,
risk, high
yeah,
rave basket. So
yes, that's
yeah,
right. Yeah,
totally agree with that. I think I'm probably the same way as you, Donna. I'm also a little bit more conservative as well.
yeah, yeah.
Lastly, what do you like most about Drug Hunter?
So, when I was in industry, I knew everything that was going on in my company, in the other companies. So, felt like a community. In academia, you know what's in your field, but it's really hard to keep up to date with what's going on, even important stuff, right? And so, Drug Hunter lets me kind of keep my finger on the pulse of really what's going on in drug discovery that I wouldn't have a mechanism to do otherwise. And so perfect example is we don't work on RAS, but the reason I know about what's going on RAS in the RAS world is the stuff I see from Drug Hunter. And so thank you for that. You kind of keep me up to date.
That's amazing to hear. Yeah, thank you so much, Donna. We really appreciate that. That's kind of exactly what we're there for. And yeah, so before we dive into Donna's presentation, I just want to briefly show folks a new feature, a new category that we just recently implemented at Drug Hunter. So if you go here to the full text search and search for targets, we actually have two new categories of reviews on targets. So there's the full target review, where we, in this case, it's CDK2, where we can look at the competitive landscape. We look at a few structures, even undisclosed structures here as well, where, you know, the structure is not known yet, but maybe some of the data associated with that molecule is known. We talk a little bit about the biology. Why is this target so important? You know, what makes these assets individually important? Then, you know, go a little bit into detail about each asset. And even if the structure is undisclosed, we oftentimes go dive into the patent and extract a structure that matches kind of the data that is disclosed about this compound, so that you can kind of know as early as possible how a potential candidate could look like. And then just very recently, we introduced this category of a target brief. This is our first target brief here. And this category serves the purpose of following a target early when there maybe is not even a lot of chemical matter that is in that competitive landscape, but when something is really heating up. So here, back-to-back Nature papers from AbbVie and the Broad Institute. Then there was a Tango bioarchives disclosure and a poster at the AACR as well. So here we're talking about the biology. Here in this particular case, it's about a synthetic lethality target, which is super interesting. And yeah, we talk a little bit about the biology so that you can quickly get an overview of what that looks like, about specific data maybe associated with the compound, and about a little bit of an outlook of what essentially what is going to happen in this particular field and what you should be watching out for. And with that, I will stop sharing here and I will hand it over to Donna. I'm very excited for this presentation, Donna, on academic drug discovery with a focus on kidney disease and Duchenne muscular disease. So,
Great.
the floor is
And
yours.
thank you, Dennis. I appreciate that. So exciting to be here. It's great to be here. I appreciate the invitation. And I know we talked a little bit that it's not frequently that you have academic scientists talking at some of these flash talks. And so what I thought I'd do in the beginning is just give you my perspective on some of the differences. And again, this is my perspective based on my time at Roche and at Wyeth, and then more recently in academia. And so some of the things that are really different are the projects that we work on. And so in industry, we really worked on moderate to low risk projects, compounds and targets that were pretty well validated before we sort of started the medicinal chemistry. And then we had very specific pathways to how to get there, right? So you had your in vitro studies, right? You did in vitro ADME, you did a PK study, and you would always do a PK study before you did your efficacy study. So very traditional, but important pathway to follow. And that was all necessary because you needed to find a drug candidate, right? That was the goal of that project. In academia, it's really different. And so you have an opportunity to work on high-risk projects, and maybe I would say even an obligation to do that. You can use unconventional approaches. You can take risks. I'll tell you that I'm embarrassed to say that we do plenty of in vivo efficacy studies without PK, just because of the resources, the timing, and some other things that I'll talk about. But the difference is that in academia, our goal is to answer a scientific question. And so if your hypothesis is that a certain target is going to be useful in acute kidney injury, if the answer is no, that's a success, right? So you can say, I answered a scientific question, then you can move on. And I think a perfect example of this is the amyloid hypothesis for Alzheimer's, right? And so there was compelling evidence that if you could inhibit beta secretase or gamma secretase, you could have disease modifying effects in Alzheimer's disease. And we still don't have a small molecule that does that. And so I think if you're in industry, that might be considered a failure. In academia, we've had a hypothesis, we tested the hypothesis, right? And now we know it's probably more complicated, way more complicated than just inhibiting one enzyme in a very complex disease system. Another big difference is the time frame. In industry, we had really short, very milestone-driven specific goals. In academia, we have really long time frames. And so the projects I'll talk about today probably have been going on at least 10 years, if not longer. Some of the reasons for that is because the financial resources we have are less. And so it will take, if you have grant funding to do something, it may take, that grant may be for 3 years, maybe for 5 years. And then often there's a gap. So it's just more challenging to make things done more quickly. And certainly in industry, you usually have especially in big pharma, you usually have all the resources you need. That's not an issue. I think another really big important difference is the access to expertise you have. And in industry, we had this incredible breadth of capabilities. So you had the med chem people, you had people who could do process chemistry like amazing. You had in vitro biology, you had in vivo biology, and everybody was working on the same project. We don't have that in academia, or in most academia places. You don't have that breadth of capabilities that can move drug discovery projects, through all the different pathways or all the different processes that are required. But what we do have in academia that we can take advantage of is like an incredible depth of expertise. And so the world's expert in a specific disease, the world's expert in protein. And one of the projects, the DMD project I talked to, my collaborator basically was one of the first to identify the protein. And so you could really take advantage of that, that you probably isn't in industry. My sense is that they broadness, right? And being able to do a lot of different things is good versus doing the depth of things. And so that's another big difference. And then finally, the most obvious difference is the mission. In industry, the mission is to generate a commercially viable drug. In academia, our mission is to educate students, right? And to generate some new knowledge and then to share that knowledge. And so our primary mission is not to get a commercially viable drug. If we do that, that's great. But sometimes the mission of educating students and really sharing the knowledge might be a little antithetical to keeping things private that you maybe for IP reasons. So there is kind of this balance and sometimes a little tension in saying, all right, we need to publish it. But we also want to make sure that we have strong IP position if we really want to think about the possibility of having someone develop this drug further, or this compound further. So sort of with that in mind, I kind of think of drug discovery as this continuum. And, I really highlighted the two terminus, but it's more of, people fall in between either one. There are certainly academic groups that have tons of resources that can do, PK studies, do GMP synthesis and things like that. And there's certainly pharma and biotech companies that do things that are high risk, very specialized. And so I don't mean to say there's two different things. There's definitely continuum. But what I thought I'd talk about today are two projects that we work on in the lab that's really more towards this one end of that continuum. Very briefly, I'll talk about our Duchenne muscular dystrophy project. This is in a really early stage, and so I'll just sort of show you how we got to where we are, and then hopefully I'll spend more time on our kidney project. So Duchenne muscular dystrophy, it's a very serious muscle wasting disease that affects young males. And it's caused by mutations in this DMD gene. And that gene codes for a protein called dystrophin. And dystrophin is incredibly important for muscle integrity. It links the extracellular matrix to the cytoskeleton, and it basically acts as like a shock absorber. So when your muscles are contracting and relaxing, they sort of absorb some of those changes, and they keep your muscle healthy so that you can continue to do that. So when you have muscular dystrophy, especially Duchenne muscular dystrophy, you often have an absence of this protein. And so you have muscles, but when they sort of contract and relax, they start to get damaged because there's no shock absorber there. And that damage eventually causes inflammation, the muscles fail, and then you get very serious consequences of those disease. It's a rare disease. There's only about 15,000 individuals in the United States, but it's a very, very serious disease. So boys are usually in a wheelchair by their early teens, can be on a ventilator by the time they're 20, and the disease is often fatal by their fourth decade. So very, very serious disease caused by a mutation in this gene for dystrophin. So there are approved therapies for DMD. One is exon skipping. These are antisense oligonucleotides that basically skip over an exon and allow a small truncated version of dystrophin to be formed in these patients. Those are for very specific mutations that occur in about 30% of the patients. There's a gene therapy approach that delivers, again, a sort of microdystrophin gene through an AAV vector that has been approved. And then there are the other therapies that are approved are corticosteroids, which reduce the inflammation. And givinostat, an HDAC inhibitor, was also recently approved. And that really reduces some of the inflammation in these fibrotic pathways that result in the muscle wasting. But despite the fact that there are approved therapies, there's really serious limitations to them. So for exon skipping, as I mentioned, it's only useful in 30% of the patients. So 70% of the patients can't use this therapy. In addition, most of these patients have never seen dystrophin because there's no protein formed. And so often it can be considered as foreign by the body, and you get an immune response to it. This happens not only exon skipping, but the gene therapy as well. So you get these immune responses that are really challenging for the patients. In addition, gene therapy, there have been some adverse events with the AAV vector as well. And then certainly it's well known that some of these corticosteroids have significant side effects that can limit the patient's ability to take them, and they're certainly not DMD specific. So one approach is based on this protein called utrophin. And utrophin is a homolog of dystrophin. It has a very similar structure to it. It does many of the same things of dystrophin. It's upregulated during development, the perinatal period. But once you're born, you have very, very low levels of utrophin formed. And so why that is, people aren't sure, but you really don't have it. But there are multiple mechanisms of why there are so little utrophin formed, but one is sort of highlighted in this slide. There is a series of microRNAs that then bind to the 3' UTR or the 5' UTR of the utrophin gene and basically cause it to that transcript to either be destabilized or degraded. And then that results in just much smaller amounts of the utrophin being formed. And so that's one mechanism by which that happens. And we think it's probably a major mechanism. So there is compelling evidence in animal models that if you can increase the utrophin levels, you can ameliorate the DMD phenotype. This has been in transgenic animals. These are in animal models. The antisense oligos can do this, and protein and gene delivery methods. So I apologize can you hear the sirens?
I can actually. Zoom does a great job in suppression of noises.
All right. Sorry about that if other people can hear it. So, but importantly, utrophin is not foreign because we've all seen it before. And so there's no immune response. And it would be effective that targeting utrophin, upregulation of utrophin, would it be effective in all the DMD patients? It wouldn't really matter of the mutational status. So how would you upregulate utrophin? One approach is to open the throttle, right? And sort of at the transcriptional level, see if you can increase the transcript for utrophin. And that has actually been done. And so the Kay Davies lab, and then together with Summit Pharmaceuticals, developed a small molecule called azutramid. It was from a high throughput screen, and it really focused on targeting the promoter, so you would have more transcript formed. Compound entered phase 2, really beautiful recent work identified its mechanism of action as an antagonist of the aryl hydrocarbon receptor, which is a transcription factor. And so that makes sense. And unfortunately, while the early results were promising. The sort of final results were not. And azutramid was, clinical trials were suspended. And so for whatever reason, it was not successful. I see in the literature, other people, they're trying to make improved molecules, maybe with better PK, maybe increasing the levels of utrophin approach achieved a little higher than the original. But so that's where that is. So our approach was a little different, and it was, instead of opening up the throttle, it was releasing the brakes. And this just shows sort of the schematic of what we were hoping we could do, is if we could find small molecules that could prevent these microRNA from binding to these UTRs, we would then get sort of this post-translational upregulation, and then get increased levels of utrophin formed. And so my collaborator, Teji Karana, who's been working on utrophin since he was a graduate student, developed this really relatively simple assay. They basically made a gene, put luciferase that was flanked by the 5' and 3' UTRs of utrophin, and then basically put that in a muscle cell line. And then we screened for small molecules that could increase the levels of, we saw luciferase in these labs. And so based on that, we also had we had a control that did not have the UTRs present in these, but they had the same promoter and luciferase. And so between the difference of those two gave us our hits. And so first we did a library of known bioactives. And we found a small molecule, TSA, which is a pan-HDAC inhibitor, interestingly enough, that had really potent activity. It increased utrophin levels about 4-fold in cells. And here's an example of where we went into an MDX model with no PK, but we have the capabilities to do this MDX model, which is it's a 3-month model. But what we saw was significant increase in RNA and protein of utrophin. This is the treated samples, and this is the untreated samples. In the three different mice, you could see basic on Western blots, you can see increases in utrophin in their muscle cells. And then we also did basically behavioral tests of these molecules. Just because you have more utrophin, does it mean that the muscles are stronger? And indeed, we have evidence that it is. So the rotarod test, which basically is a measure of kind of strength and muscle stability, we see increases, improvements in there. And then we isolated the muscles, and then there are multiple ways to measure the strength of those muscles. And we see significant increases in all of those ways strength. So very promising support that upregulating utrophin with small molecules can have this effect. Now, TSA is not going to be a drug. In fact, I was shocked that givinostat was approved for DMD patients, but it's a very serious disease. So what we did now is we expanded that HTS because we had some confidence that we could find small molecules. And we now have multiple actives. We did about 100,000 compounds. We have multiple series of compounds that have EC50s in the screening paradigm, of most of them less than 1 micromolar. And then we can see a differential between the screening and then the counter screening. And so we believe they're selective for utrophin upregulation. Western blots of these small molecules, we see effects at 500 nanomolar on some of these, and they all have pretty decent drug-like properties. It was a pretty high quality library. And so this is where we are with this project. It's, again, going on for about 10 years, more or less. But, it's been a slow project. We were fortunate to have funding for developing the assay and doing the original high throughput screen from the NIH. And then more recently, we had funding at Penn from the RNA Institute here. And so we're grateful for that. And currently we are working on, slowly with very little resources, the SAR expansion optimization. But really interesting would be confirming the mechanism of action. Is it really binding to RNA? So I think I should also just want to mention one more thing. We also have another project where we are using antisense oligonucleotides that would also block. We had funding from NIH to do that, but sadly, in the middle of that grant, which was going great, it was one of the casualties of the recent cuts at NIH. And so we got a letter that said, your grant has been stopped. And so So that was disappointing, but I think it's really promising, and hopefully we will find other ways to pursue these really exciting and potentially impactful approaches. So I'll spend the rest of my time talking about our project on AKI. And so acute kidney injury, which I didn't know about when I first started this project, is a sudden temporary loss of kidney function. It's unlike DMD, it's really common. And so if you are in the intensive care unit, there's a 50% chance that you will have an episode of AKI. And it can be very serious. Some people recover, but some don't. Some people actually can succumb to it. Some people need dialysis. But importantly, it puts you at a much higher risk factor for developing chronic kidney disease and end-stage renal disease. And when you have that, now you're on dialysis or you're on a list for a transplant, right? So again, very serious. And that's thought to cause because that acute episode may leave you with scar tissue or fibrosis on your kidneys, and that's what leads to the chronic kidney disease later. So it's an unusual project, and I think because what we're hoping to do is treat during this acute stage, but then have an effect 10 years later, and so patients aren't developing chronic kidney disease. And so what do you do if you have AKI? And so if you look in Wikipedia, it tells you you really try to prevent cardiovascular collapse and death, and then you call the nephrologist. So there's really no drugs to prevent it. Certain patient populations are prone to it. There's no way to treat it. There's no way to accelerate the recovery, and there's certainly no way to prevent that post-injury fibrosis. And so why is that? I think one of the reasons is clinicians and scientists were very reluctant to biopsy failing kidneys and really understand the pathophysiology that was going on. There's no prognostic biomarkers, and so you can't say, yep, these are the patients who are going to get it. These are the patients who not. So clinical trials could be really challenging. Patients present late in the disease, their kidneys are already failing when you see them or when you realize it. And then there are a multifactorial condition. So diabetics get it frequently. You can get it from taking certain antibiotics, certain anti-cancer drugs. And there's really no validated target. Nothing that say if you target this protein, you will definitely have an effect on AKI. It's just the science isn't there yet. And there have been multiple clinical trials failures. And so it's really challenging, but I think it's a perfect academic drug discovery project because it's high risk. There's really a lot to be learned from this. The clinical trials could be really challenging to do. And the ideal endpoint is not after the acute account, but really 10 years later, are people not getting CKD? So this was a project that was very disease first, and my collaborator, Neil Huckrady at the University of Pittsburgh, here he is, a developmental biologist who specializes in zebrafish, and there's the zebrafish tanks at Pitt, and he did a screen of a small number of compounds, about 800 compounds, in an assay where you take zebrafish embryos, you inject gentamicin, and that causes the kidneys to be damaged just like it does in humans. Then we treat with compound, and then we score survival. And so if you don't add anything, most of those embryos will die. And we looked for compounds that improve survival. And so he came up with this incredibly, we saw calicheamicin, and this is like the opposite to calicheamicin. It's an incredibly simple molecule, phenylthiobutanoic acid, that had this effect. And just here's what these curves look like. This is a survival curve. So if you don't treat the fish after the gentamicin injection, basically most of them will not survive after about 4 days. If you treat with phenylthiobutanoic acid, you see over 60% survive after 7 days. And so it is remarkably effective. Except it's a carboxylic acid. And so while it worked in the fish assays, because literally the fish were swimming in it, it didn't work in higher animals. And so we had to do a prodrug approach to see to validate it. And so we made a series of prodrugs. We made the methyl esters, we made this benzimides. And what we can see is in all cases, this is the chart for the benzimide. You see rapid disappearance of the prodrug, right? And then a concomitant appearance of the carboxylic acid. And so here's an example where we actually did the PK study before the efficacy studies. And so it's not always that we adopt that, right? And these also work in the zebrafish as well, so that zebrafish are metabolically stable or metabolically active. And so we could put this compound in mice, and this was done by our collaborator, Mark DeCastro at Vanderbilt. And in this model, basically you take one of the kidneys, you basically clamp it, and then we treated with 3 different regimens of this compound we call 186. Then you remove the other kidney. And so all the then kidney flow is through the damaged kidney. And we measured things like creatinine, BUN levels. And then, 28 days later, we look for markers of fibrosis. And so doing that, we saw really in 2 of those dosing regimens, significant reduction in creatinine. And creatinine is an important marker because it's one of the things they measure with patients, with human patients in the hospital. And then those same 2 dosing regimens, we saw a significant reduction in fibrosis. So hopefully that, now if this was given to a patient, they would be much lower risk for chronic kidney disease. And so importantly, also the compounds are efficacious after the injury, which is how patients are going to see it, not pre-treatment. We know that the conversion to the acid is required for the efficacy. We had made compounds that can't be converted to the acid and they're not effective. And we have multiple models. We've put this compound in multiple AKI models. And so not only sort of this very physical injury, but injury with things like cisplatinum, other molecules. And so it's broadly effective. So great, we have this prodrug, and as we all know, it's a great way to fix permeability, other flaws, and in fact, about 12% of approved drugs are prodrugs. But for kind of a small academic group, prodrugs are really challenging. You may increase the permeability, but your solubility is going to go away. But more importantly, it's really hard to predict the metabolism that's going to release the active species. And there are certainly differences between the species and those metabolisms. And so, now we're working in fish. What's the metabolism in fish? We'd have to go to mice. Eventually, we'd go to higher species and then humans. And so it would have been really, really challenging. And we just didn't have the resources to do that kind of development. And so our goal was really to find a compound that was active as the parent. And so didn't need the prodrug. So fortunately, and I'll get to that, but sort of the approach was, can we find an isoster that replaced the carboxylic acid but maintain the efficacy of the prodrug? And the question was, which acid isoster should we use? I know you had Nick on here a little while ago, who's Mr. Isoster. But because at the time we didn't have a lot of information about how this was working, we certainly didn't have an X-ray structure. We don't know exactly where these compounds bind. We made a couple of assumptions. One was that histone deacetylase 8 was the likely target, and I'll show you data later about why we think that's the case. And then we assumed that the pKa was important for target binding, and so we wanted to match that. And then we also wanted to monitor lipophilicity and permeability. And so a number of years ago, we had published this paper where we, for a different project, we were really interested in the properties of carboxylic acid isosteric replacements. And so in this paper, we made a series of these bioisosteric replacements on the same backbone and then measured their properties. And so it's a great database and it's a great resource. And so this slide shows the carboxylic acids in that paper and the isosters. And then here is the parent carboxylic acid, and then everything is related to comparing to the carboxylic acid except the pKa. The pKa is on an absolute scale, so it's color-coded. So everything with sort of this buff region, buff color is the same pK as the carboxylic acid. But then apparent permeability based on a Pampa assay, everything to the right on this slide is more permeable than the acid. To the left is less permeable, and then log D at 7.4, everything above it has a higher log D than the acid and everything below it. And so we use this because, again, we had limited resources to, rather than sort of randomly pick what to make, to try to be very selective and rational in what we made. And we really focused on compounds that had similar pKa's, and then hopefully lipophilicity in the same range as either the prodrug or the acid. And so we only made a handful of these isosteric replacements. Some of the ones we've shown were here. And we kind of tried to match the pKa's log P's. They were compliant with Lipinski's rules and some other rules that we developed that would be effective, predict whether they would get into zebrafish. And we right away found small molecules that were effective in our fish model, although really high doses, right? So this is not really that useful. It just gave us confidence that we could find an isosteric replacement for the carboxylic acid. But the winner was the acyl sulfonamide. And so this compound is as efficacious as the prodrug in our zebrafish survival assay, and that's shown here. This is in the green, and this is the prodrug, and so it works well there. We've been working in fish. People kept saying, how about humans? And so Neil developed a kidney organoid assay where he generates organoids, and then we damage with hemin, treat with compound, and then we analyze for fibrosis and other markers of inflammation. And the compound, at different doses, we can talk about what the kind of interesting dose response curve, but kind of at relatively low doses, 200 nanomolar and 1 micromolar, we see significant reductions. This is a marker of kidney injury. These are markers of inflammation. And so we see them going down in most cases. And then this is a marker of fibrosis. And so we see, importantly, these reductions in fibrosis at pretty low levels in, a human model. And so that was also really encouraging. And then we did go into a mouse model with this, again, very similar to what I talked about before. We measured BUN and glomerular filtration rates instead of creatinine. We didn't see huge changes in BUN levels. We saw some significant changes in GFR, glomerular filtration rates, at 5 milligram dose. And at that same dose, we see the reduction in fibrosis as well. And so in this higher animal. And so so we're really excited about those molecules, and we continue to work on that. But really unsatisfying is the mechanism of action. What we see is that when we treat with these molecules in the zebrafish, we see a significant accumulation of an HDAC8 substrate, acetylated SMC3. And so here's what happens when you treat with the compound. Here's the increase in that substrate when you have the HDAC8 knockout. And so it suggested that the molecules are HDAC8 inhibitors. We have some other evidence, but interesting, if you put it in an isolated enzyme assay, it's not active. And so it sort of remains to be seen how these molecules are working. And we have some other evidence that perhaps they're allosteric inhibitors, or HDAC8 is not a bona fide, some people don't even think it deacetylates histones, that it probably has other substrates, that it has other function, it has some scaffolding functions, and so perhaps that's how it's working. But another aspect of the project is making probes based on those molecules to see if we can pull down exactly what it's doing. But to validate that HDAC8 was a target, Neil generated fish that either were heterozygous or homozygous for HDAC8 knockout, and you can see the complete knockout we see are really resistant to this damage, this kidney damage, really supporting that HDAC8 is important for the recovery that we're seeing. And then an interesting study is we took a suboptimal dose of the prodrug, and that's shown here, and really has no effect in the wild type. But when you add a suboptimal dose of the compound with the partial knockout, we now regain activity. And so again, it supports that these compounds are working through HDAC8. And that led us again to think, all right, if they're really working through HDAC8, there are plenty of reported active site HDAC8 inhibitors in the literature. So we went there, we pulled a bunch of those out, and indeed, many of them have, allow a survival in our fish model. And so here are 3 different molecules that we pulled from the literature that do this. And then PCI 3401051 is kind of a standard, very potent selective HDAC8 inhibitor. That compound we put in our mouse model of AKI. And again, we see at certain doses significant improvements in fibrosis and glomerular filtration rates. So supporting that, if you target HDAC8, regardless of whether it's active site inhibitors or this other way, which we're really not sure of, you will have an effect in AKI. And so we are actively working on this project. We fortunately have not had this grant canceled, but we're looking at improving the potency of those compounds. And I think we need to get a little better exposure. And then asking the question, how are these really modulating HDAC8? It's an interesting question because there's very little, there's not a lot known about allosteric inhibitors of HDACs, or certainly HDAC8. And so I think that would just in general be an interesting question to answer. So just in closing, right, I hope shown you the types of projects that I think we can have an impact on in academia. The AKI project, we didn't have a validated target to start with, but we think we do now with HDAC8. We've been working on these projects for a really long time, but both of them really relied on some depth of expertise. And so certainly Teji Karana, has expert in utrophin and DMD that we really rely on. The AKI project, our previous work on isosteres and prodrugs has really helped that project. And then my collaborators in developmental biology who know everything about fish and AKI models. And as I mentioned, these projects were supported by grants from the DoD and the NIH, and DMD from NIH and the RNA Institute. And so I think we've educated some students. Hopefully we're sharing some knowledge, including today. And I just want to thank the people in the lab here, my current lab, but Neil at Pitt, Mark at Vanderbilt, and Teji at Penn in their labs, and Dave Schultz, who did the high throughput screen, and then the chemists who worked on this particular project in the lab. So.
Awesome. Amazing. Amazing, Donna. I learned so much about this. This is really, really super cool. I had a brief dabble in DMD during my industry career, so I know it's a super interesting and yeah, definitely underserved patient population.
Yeah, it's underserved, and there's like 6 drugs approved, but they're just not great.
Yes, yes. Yeah, I guess one question that I had would be Givinostat. You mentioned earlier Givinostat as an HDAC inhibitor. I believe it was approved last year, and I was equally shocked about this as
So,
well. It doesn't even have a black box warning. I actually just looked at the label while you're talking. Do you know if anything is known whether it's a pan inhibitor, like
I think
how?
it's a pan inhibitor. I think maybe it's towards class 1.
Yeah.
I think generally it's a pan inhibitor. The HDAC literature is really challenging because there's a lot of debate about selectivity of HDAC inhibitors. And there are papers that say some of them are slow binding inhibitors, and so they appear to be selective, but they're actually not.
Interesting.
And so it's kind of fascinating. So I don't, but I think it's more pan.
Yeah.
And, the disease is so devastating that I think the bar is a little lower.
Yeah. Yeah, makes sense. Have you or anybody else that you know of looked at whether Givinostat is also an utrophin upregulator?
It came up in our screen.
Oh,
Yeah,
it did.
it
Yeah, yeah, yeah,
did.
yeah.
Yeah. And so what we're thinking is that there's sort of 2 different activities, but it did come up in our screen, not one of the sort of highest ones, but it did come up.
Sounds great. Cool. Yeah, let's go through a few audience questions here as well. So Yoshi is wondering, how do you pick targets or projects in academia as compared to industry? And if you had to brainstorm for a new target, where do you start? So in industry, we might look at new protein targets and pathways we already have experience in, or at least indications we've worked on before. But how is that in academia?
I think it depends. In my lab, we try to find projects that we think we can have an impact on. And so, if someone comes and says, I have a new kinase, Donna, I'm like, I don't think I can do that. That's right. Just because we don't have the resources to kind of compete in that area. But other people have done a fantastic job in academia working on kinases. So something we can have an impact on. I like to work on projects where we have some expertise. And so, we work on a lot of projects where we want to they start with a carboxylic acid, and we think our expertise, or at least our previous work in carboxylic acid isosteres, can complement that. And so we have a couple of projects like that. One, another rare disease called Friedreich's ataxia, where oleic acid was found to be protective. And so we made a series of isosteres and found really interesting compounds doing that. And it is, I think the other thing is we work in collaboration. And another criteria is that we have really good assays, right? Because that's what medicinal chemists need good data.
Yeah, no, that's a really good point. Well, in your graph about the carboxylic bio or acid bioisosters, I was wondering, you ideally want to be in the bottom right corner, right?
Yes, you
More
do.
permeability and less lipophilic, but I didn't see any compound in there. So
Yeah, no, we really wanted to be in there. And I think some of the ones with the PKs were not there,
yeah,
right? And so we needed kind of PKs in the right range. And then I think so that's just should be characterized just the carboxylic acid part of it. But the molecule has other pieces to it. And so that changes, our molecules are more lipophilic than what's shown on there because they have this phenyl propyl chain on it.
now. Yeah, sounds great. And I guess we have a few more audience questions here. Yoshi is wondering, at one point if I heard correctly, you mentioned studying metabolism in fish, and he says he doesn't have experience with that. And if you could describe the pros and cons of, drug metabolism in fish as opposed to maybe other species, that'd be super interesting as well.
Yeah, so so we would talk about we were doing the study in fish, and I would say to my collaborator, like, I don't know what Lipinski rules for fish are. I don't know how to predict what compounds are getting in and what compounds are not. And there wasn't. We actually tried to do studies. We tried to get fish. Actually, what we tried to do is convince someone who had uh, a fancy mass spec, LC mass spec, to let us do these studies. They did not want to put smushed up fish on their LC mass spec.
Yeah,
Um,
I can.
yeah, so what we did instead was we basically just we did a literature search of all the compounds that were reported to be active in a zebrafish assay, embryonic assay. And then we took those structures and then we just did an analysis of what are the properties, just like Lipinski did, and said, what's the average log P? What's the hydrogen bond donors, acceptors, molecular weight? And sort of we just came to, it actually turns out they're more lipophilic than traditional compounds, and they have a sort of a narrower range than Lipinski rules. And so it just helps us think about if the compounds are going in zebrafish, we don't want them to be too far outside of that range, because we don't know whether inactive compounds just didn't get in.
Yeah.
And then, yeah, and I can go on and on about zebrafish, but I won't.
The dosing is probably complicated too. I mean, yeah, I guess you dose in the tank or something like that. You said they're swimming in the acid. So it's kind of interesting, yeah.
Yep. And that certain concentration, that's what they're swimming in. And and it's a tough assay. It's kind of low throughput. And so we
Yeah.
really, all those assays, we just do at one concentration.
Yeah.
And so, that you get very low resolution data from that.
Makes sense. Makes sense. All right. Jesse Simon is wondering, in your opinion, how can we encourage more academia industry partnerships to leverage the benefits of both sectors?
So one of the things that I think is changing that I'm really excited about is that I do see more academia industry partnerships. I see that more in terms of synthetic chemistry. They've developed new methods that, maybe they partner with someone in academia to develop some methods that then is generally useful. I see it less in kind of drug discovery, and I think that's because there's the ownership issue. And who owns the molecules and who's going to get paid. And for better or worse, the Hatch-Waxman Act, right, encourages academics to patent their work. And the patenting offices have seen these amazing successes in academia, right? Dennis Leota, Richard Silverman, and so they think every little molecule you're making is going to make them $1 bazillion, and they're not really, right? Which is not reasonable. And so they're my experience is that these offices are very, very conservative about sharing potential for intellectual property, because they see the potential and they don't want to give it up, even though, it's kind of not great likelihood, but, and so I just see less of that. And I think it's disappointing. Um,
Yeah, yeah, makes sense. We have another question here about the DMD model. So how does the level of dystrophin compare between normal and treated subjects in the DMD model? D does,
so
yeah, oh yeah,
Yeah,
maybe.
so what we see depends on where you look. So we see about we see over twofold increase in utrophin over normal levels. And we see when we look at, depends on which levels you look at, mRNA, protein, but typically we see between 2 and 4%. And 2 and 4-fold difference,
Fold,
right?
fold, yeah, yeah, yeah.
Fold difference. And so I think one of the questions is how much do you need to see,
Right,
right?
exactly.
And so one of the ideas about why ezetimibe failed is that it was a twofold increase in utrophin levels. And so the question is, was that just not enough? And you need more of that. And so, in our workflow, we're really looking for compounds that give us more than twofold, assuming that it has to be at least twofold because twofold didn't work in patients.
Makes sense, makes sense. So yeah, fourfold, maybe fourfold.
Yeah, maybe fourfold, I don't know. That's sort of an unknown, especially in humans, right?
Yeah, yeah, makes total sense. Yeah, so let's talk maybe a little bit more about AKI. Actually, I have to admit, my dad had an acute kidney failure early last year, which was very, very, very shocking. And here's a question from Chao Cheng. Other than acute kidney injury, do you think HDAC8 inhibitors might be working in some other kidney diseases like polycystic kidney disease?
In fact, I know the answer to that. In fact, they do. Right.
That's
And so
amazing. Yeah.
yep. And so we have we have a grant that focuses on these same molecules in HDAC8, and they look to be effective in follicular glomerulosclerosis, so FSGS models. We haven't looked at polycystic kidney disease, but I think that our goal is to get them into as many models as possible. But certainly it works in FSGS very well, in fact.
Yeah, that's great to know. I mean, yeah, so it seems like we should be looking at more HDAC inhibitors, and I think people are
Yeah.
wondering in the questions as well. So Benjamin here is wondering, is there any effort ongoing to obtain crystal structures with HDAC8? I believe there's some in the literature for the compound you showed.
Yes, and in fact, my collaborator, literally the floor below me, David Christensen, has published and solved multiple HDAC X-ray structures,
Yeah,
including HDAC8. And so the first thing we did was gave him a vial, and we didn't see any binding to isolated HDAC8 by isothermal calorimetry.
interesting.
And so for whatever reason, when HDAC8 is isolated, we don't see effects of these small molecules. So it either is inhibiting maybe a protein-protein interaction that it's important for some of its other effects. It's an allosteric inhibitor where you just doesn't have great tight binding. On good days, I say, oh, it's a glue.
Yeah. Yeah, I think I saw some paper talking about the molecular dynamics. They also didn't have a crystal structure, but I think they looked at it with native mass spectrometry
Yes,
and
yep.
molecular
Yeah, yeah. So
dynamics.
we're trying to do some of that to see if, can we get can we find the partners or what's the complex? And HDAC8 is part of a bigger complex when it's active. And so, somehow it's working with some of that. So I would love to get, we're making these activity-based probes, and hopefully we'll find, some evidence of the complex that it's binding to.
Yeah, no, that's super interesting. And you mentioned that, HDAC8, some people believe that it doesn't only deacetylate histones. Are there, like, in your experience or from what you've been reading, any other client kind of proteins that HDAC is deacetylating or doing something else too that's
Yeah,
not
and
even
so
deacetylating?
so SMC is a protein, and that's part of this cohesion complex involved in sort of mitosis, I think.
Yeah,
So it has these other substrates. There's a huge, effort of people trying to figure out what the native substrates of HDAC8 are. And so, I think some of like the Furky lab has done some of that, but there's been a lot of work trying to find out what they are. Just because it deacetylates a protein doesn't mean that's what it does naturally, right?
Yeah, yeah, absolutely. And maybe I think we're a little bit over time, but maybe as a last question, um, do you have maybe like a comment on common side effects or toxicities of maybe pan HDAC inhibitors versus selective HDAC8 inhibitors?
Yeah, and so that was one of, it's a big concern.
Yeah.
And so certainly there is concern about hydroxamic acids and very strong zinc binding groups. It's one of those things that are lore, but people are worried about, or we would be worried about sort of non-specific binding to metals or metal-containing proteins that those classic HDAC inhibitors would have. Certainly pan inhibitors would do that. And so the beauty, why we think HDAC8 is because it's not a traditional histone, it may not have histones as substrates, then it may not have the same side effects as some of the others, these pan inhibitors. So,
Yeah,
but there's
definitely.
also, and you said javinostat didn't have a black box, but most of the other HDAC inhibitors are mutagenic, are Ames positive.
Oh, interesting. Ames positive. Yeah, I was thinking about I think thrombocytopenia side effects,
Yep,
neutropenia,
yep,
and anemia in some cases. But yeah,
yep. And
I was,
yeah, and you see on our graphs, right, at higher concentrations, we see what we think we are seeing is some toxicity, right?
and
So we have these curves that aren't perfect dose response curves, but we think we're seeing, so just it's not enough of a window yet on some of these compounds, that we need to have more selective compounds.
yeah, yeah, makes total sense. Now, I was especially shocked because that's going to be chronic dosing for DMD, right? So, uh, yeah,
For
that's
children, for children.
children, exactly,
That's
pediatric
right.
indication as well. So, I think
Yep.
Givino said what, if I look correctly, the dosing can be started for patients that are over 6.
Yeah. Yep.
Yeah. Yeah. Well, Donna, this was amazing. Thank you so much. I
Thank
learned
you.
so much about DMD and yeah, AKI. That was super, super interesting. So yeah, again, thank you for the great talk. Thank you to the audience for the engaged questions. Really, really always enjoy this Q&A session. And yeah, we will be back here next week with Renata Obala from Chinook, a Novartis company. And she will be talking about the discovery of CHK336, and that's a lactase dehydrogenase inhibitor. So I'm also going to be very excited about that. And yeah, with that, Donna, again, thank you.
You're welcome. A pleasure.